Hi, I'm Dr. Vivian Lowe, and you're on VLMD Rounds, a podcast on medical science and tools to optimize your health. Today, I had initially planned to talk about the immune system in its entirety, and then when I looked at it and started to prepare my notes, I realized, ooh, that's a lot to do in one session. So I decided that I would just divide it into two parts. This episode will just focus on one side of the immune system, the innate side, and then at a later episode, I'll go into the adaptive side of the immune system. Because, as I said, it's such a huge topic. And again, today's episode, we're just going to paint a very broad picture of the immune system. Okay, I always think of the Mahabharata when I think of the immune system and all the players in it. And that's an ancient Indian epic text with, just thousands of characters and very voluminous, very hard to remember everyone and the different stories that are in there. So when I look at the immune system, it often makes me think of the Mahabharata in that sense. All right, so let's go. Now, with regards to the immune system, as I said earlier, we think of it generally in two parts. One is the innate immune system, and the other part is the adaptive immune system. So the innate part is pretty much your quick response to any infection or any danger or damage to your body. For example, if you cut yourself, well, you can't wait around for a few days to get an immune response. You really need to have your immune system kick into gear as soon as possible. And so that's the innate side of the immune system. The drawbacks to that would be, yes, you do have a quick response, but it's not going to be very specific. So it's going to be a very generalized response. We're going to realize, oh, I think we're being invaded by some bacteria. We don't quite know too much about the bacteria, but it's not a good thing and we need to defend ourselves, okay? So that's a general picture there. The adaptive side is actually highly specific because over time, as we gather more information about the invading pathogen, then we can target those pathogens more specifically, and that will be the job of the cells of the adaptive immune system. As I said, today we will focus on the innate side of the immune system. Now, there are many components to the innate side, and I'm going to just paint a very broad picture so that you get a sense of that quick response that your body tries to muster when it encounters a bacteria or some other invader in your body. Okay, so let's start with the complement system. COMPLEMENT, that's the complement system. And this is made up of about 20 different proteins that can kill pathogens, but also signal to the rest of the immune system that there's trouble brewing. Okay, so that's a general broad function. And there are various ways that you can activate this complement system of these proteins in your body. We have the classical activation pathway, and even though it's the classical activation pathway, it's really not the oldest system, and it's activated in the classical pathway by antibodies. All right. It was, however, the first activation pathway discovered, which is why it's called the classical. An older pathway is the alternative pathway. So even though it's alternative, it's been around longer, and pretty important to have that pathway, but it was discovered second to the classical pathway, and that's why it's called alternative, okay? So with the alternative pathway, which is what I'm going to focus on, we look at these proteins that are made by primarily the liver, and the most abundant of these complement proteins would be something called C3. So that's capital C and then the number 3. C3 is, once it's released into the blood, it gets cut or cleaved into 2 fragments, and that would be C3A, small a, right, and C3, small b. Okay, so it gets into the blood, and then we cleave it or cut it, and now we have 2 parts. We have C3A and we have C3b. Now, C3b is very, very reactive, and it quickly looks around, and anything near it that has an amino group or hydroxy group, it's just going to bind to. All right, so it's a spontaneous reaction. And there's abundant C3 that gets cleaved into 3A and 3B, and then that 3B is very reactive, looks around, anything nearby with an amino group, anything nearby with a hydroxy group, let me bind to it. If it doesn't bind within about 60 seconds, then it's just neutralized by binding to water. Okay, so all of this is happening very quickly and spontaneously in the blood. In other words, for C3 to actually do its work as part of the immune system, it really needs to be in close proximity to whatever pathogen has just invaded your body. Okay, because again, it gets neutralized so quickly that it's not going to really kind of travel around the body to look for trouble. Okay, just whatever happens to be nearby that it can bind to, it will. And if it doesn't see those groups to bind to, then it'll just be neutralized by binding to water. All right, so but imagine you have now a bacteria that's nearby, and C3b is able to quickly bind to it, right? So that's a good thing. But once C3b binds to it, we set off a cascade of events that eventually lead to the destruction of the bacteria. I am just going to briefly go into some components of it, just so you get the sense of this domino effect that goes on with the complement system, okay? So as I said, C3b has now, and then that's a little b, so C3b has now bound to a bacteria. Okay, and then another protein called B, and that's a capital B, binds to it. Okay, so binds to C3b, which is now attached to the bacteria. All right, and then we have another protein. See what I mean about domino effect? So we have another protein called protein D. And protein D is going to cut B, that's bound to C3b, right? And now we're going to make something called B little b, BB, okay? And that whole complex now is C3b that's bound to B little b, okay? And that's quite a mouthful, so we'll just call that convertase, C-O-N-V-E-R-T-A-S-E. So now we have convertase. And the thing about convertase is that it will now look around, and any other C3s it sees, it's just going to cut it into C3A and 3B. So we start to have this snowballing effect and this positive feedback loop where we're making a lot more 3Bs, C3Bs, that will bind to bacteria, and then B will bind and then get cleaved, and we make more and more of this convertase, all right? Now, we can have, when convertase is bound to C3, it can cleave another protein called C5. And so now it's going to cut C5 into C5A, C5B. And C5B will conglomerate with other complement proteins and form something called a membrane attack complex, or MAC, all in caps, MAC, membrane attack complex. And this essentially punches a hole in the cell membrane, and it's finito for the bacteria at that point, okay? So you can see there's this cascading effect. Eventually, though, we cut C5 into 5A and 5B. 5B will associate with other complement proteins and form this MAC, or membrane attack complex, which will now punch a hole in the invading cell, and that will be the end of that cell. Okay, and you'll find that a lot of these components of the immune system, they like punching holes in things. Okay, so that seems to be a strategy, is just to punch a hole in the invader. Okay, now I said that C3b is very active, very reactive. If it sees an amino group, if it sees a hydroxy group, will quickly bind to it, but it is neutralized by binding to water. And there are other proteins in your blood that will deactivate your C3b, so that we don't get a situation where you just have too much of this C3b around, and then you have a hyper-reactive complement system, and that could get us into trouble, right? So there's a control loop as well, because there are other proteins that are in your blood that will deactivate that C3b. Okay, so that was one pathway, which was the alternate pathway. Now, let's talk about the other pathway that's called the lectin activation pathway, lectin, Lectin. And this, again, the liver makes a protein called mannose binding protein, MANNOSE, mannose binding lectin. So we can abbreviate that to MBL in caps, okay, MBL. And MBL, this mannose binding lectin, it can recognize and bind to carbs and lipids that are on invading cells, not on your own cells, but any invading cells, okay. Now, MBL itself is bound to MASP, all in caps, MASP. And this stands for MBL associated serine, SERINE protease. Okay, so much better just to leave it at MASP. Okay, MASP. So remember, you have now this MBL, and it's bound to MASP. Okay, when we see a pathogen around and we have mannose on the surface of the pathogen, then MBL can bind to that mannose, right? And once it does that, that MASP will basically look at any C3 that's around and cleave it, and we'll make C3a and C3b. And then when we have C3b, that can bind to the pathogen, and then we set off the complement pathway, okay? So that is the lectin activation pathway. The difference between that and the alternate pathway. Remember, the alternate pathway was spontaneous. So, once, C3 is cleaved into 3A and 3B, it'll just start the pathway. And there are proteins in your blood that do that cleavage, okay? With the lectin activation pathway, really the MBL binds first to mannose, right? And then the MASP that's bound to the MBL is going to be doing that cleaving of C3 to 3A and 3B. All right, one other thing that I want to point out is sometimes we have C3 and it can be clipped in such a way that we make inactive C3b, okay, or small I C3b. So it's called inactive C3b because it doesn't actually lead to the assembly of MAC, or the membrane attack complex, All right, so it's inactive in that sense, but it can still bind to pathogens. It can still bind to a bacteria, for example, right? And it's useful because when it's bound to the bacteria, this inactive form of 3B, it acts as an opsin, O-P-S-I-N. And opsin would be something that would just tag the pathogen, okay, for other cells such as phagocytes to come along and kill this bacteria, right? So it's acting as a tag and saying, hey, this is what you want to gobble up, okay? And that process is called opsonization when you tag the bacteria. So inactive 3B, C3B is still useful because it's acting as an opsin, it's acting as a tag to let other immune cells know that this cell needs to be destroyed, okay? Now remember when we cleaved C3 and C5, and we had from C3, C3A and C3B, and from C5 we had C5A and 5B, right? So the A portions of those proteins are still useful. Okay, we talked about the B parts, but the C3A and C5A are chemoattractants. You can just kind of use the mnemonic as A for attractants, right? So they're chemoattractants, which means they are going to be attracting other immune cells to that area. So it's very useful. They're sort of a marker, like there's a problem here and they bring other immune cells to the area. So for example, C5A is a powerful chemoattractant for macrophages, all right? One of my favorite immune cells. So just quickly, the three main things that the complement system can do, first of all, it can just flat out kill the invader or invading cell by assembly of this membrane attack complex, or MAC. Okay, so that's one. The other thing it can do is it can function as opsins, so it can tag the invading cells so other cells can destroy them, right? And then they are also chemoattractants, so we can actually bring immune cells to the area of infection. Okay, so that was the complement system. Now we're going to look at some cells of the innate immune system, and these are professional phagocytes, okay? Which means that they're really good at gobbling up any invading, cells that come into your body. So it could be viruses, it could be bacteria, it could be different fungi, all right? So these phagocytes basically are able to swallow up and eat up the invader. And essentially, once you phagocytose it by surrounding, so they develop these arms and surround the pathogen, and then those arms meet, right? And now we've encircled the pathogen and we form a phagosome, right? And that phagosome has captured the pathogen within it. It will eventually fuse with a lysosome. So we have a phagolysosome, and lysosomes are going to dump its acid and enzymes into that pool, and we're going to now eat up really and destroy the pathogen, okay? So that's the process of phagocytosis. Now, of course, I think by now you know that one of my favorite phagocytes in the body would be the macrophage. And again, I've said this on various occasions, but a quick reminder, so we don't have to go into great detail. You can look up a video presentation that I did called the Magical Mystery Macrophage Tour. And this is generally a survey, very brief survey on macrophages in the body. And really in that presentation, I mostly focused on the phagocytic capabilities of macrophages, right? So that's a good general introduction. There are many other things that macrophages do, and I didn't touch those things in that presentation, and I'll just tell you a little bit more today, but we could probably do 2 or 3 other episodes just on macrophages alone. I might do that. Okay, so macrophages, they can come from 2 main sources. You could think of it that way. One would be tissue resident macrophages, and these were really developed in the embryonic stage, and those now inhabit the tissue, and they tend to have more of housekeeping or homeostatic functions. All right, so they just go around and make sure everything's fine, and as cells turn over, because cells are born and die every day in your body. So when we have these dying cells, the senescent cells, then these tissue resident macrophages go around to do the cleanup, and they mop up the old and dying cells. So they just keep the environment nice and healthy. And then we have bone marrow derived macrophages. Really, they come from monocytes that when they enter into the tissue now become macrophages, and these tend to respond to inflammatory situations in the body. Okay, that's a very big generalization. And of course, when you talk about macrophages, I mentioned this in the macrophage tour presentation as well. People always talk about M1 and M2 polarization, with M1 macrophages being inflammatory and M2 macrophages being anti-inflammatory. That's a huge generalization and really quite an oversimplification, but it's useful when we're talking about different effects of macrophages in the body. So I think a lot of people don't like the terminology, but we still end up using it that way. Okay, but in reality, it's macrophages are not M1 or M2. All right, I'd like you to remember that there's a spectrum, and at different times and again in different contexts that they can express M1 characteristics, and also maybe under other circumstances more M2 characteristics. So it's more of this blend and a spectrum, okay, not black or white. All right, so phagocytosis is something that macrophages do very well, but when they are primed by certain signals, they also can do something called antigen presentation. All right, so before I go into antigen presentation, let me talk to you about MHC molecules. So MHC is major histocompatibility, and you might have heard of the term, especially when they talk about transplants and so forth. Okay, but very quickly, we have MHC1 molecules that the cell will display on its surface. And we also have MHC2 molecules that it can display on its surface. All right, the difference is, think of it as 2 different social media platforms, okay? MHC1, you can only post whatever has happened in your house, okay? So if you had a birthday party and you took pictures of those celebrations, you can post that on MHC1 social platform. Okay, think of it that way. MHC2, you post about what's happening in your environment. So now your neighbor had a party, right? And you saw them outside, and maybe they were barbecuing, and you took some pictures. All right, sneaky. All right, but you took some pictures. You can post that, and you would post that on MHC2. Okay, because that platform is for you to post about what's going on in the environment outside of your house. Okay, so MHC1, again, the cell can post, in a sense, what's been going on inside the cell, whereas the MHC2 molecules, the cell is going to use that to post what's going on on the outside. Okay, simply put, that's how you can think of the MHC molecules. Now, back to the macrophages, because they like, gobbling things up, they like phagocytosing different things in the environment. So let's say there is a pathogen out there and they have phagocytosed it and gobbled it up and now digested it, okay? And some of these parts, the proteins from that pathogen, it's going to take and post on the MHC2 platform, okay? And when it does that, we call it antigen presentation. So macrophages are antigen presenting cells because they can post whatever they gobbled up from the environment. They can post bits of it on MHC2, and this is very useful because they're essentially gathering information from the environment. Think of it that way. And this information, especially if we have just been invaded by a pathogen, this information is very useful. And we're going to take this information and we're going to bring it back to inform the cells of the adaptive immune system. So they're acting like intelligence here, okay, on the field. They're not only killing the invaders, but they're also gathering a lot of information so they can take it back and inform the adaptive immune system about the enemy, okay? Now, macrophages have receptors to pick up all kinds of different components in the environment. So they have receptors for LPS, or lipopolysaccharide, and this is it's an endotoxin that's in the outer membrane of certain bacteria, what we call gram-negative bacteria. All right, you always hear about LPS, all in caps, and that's really what it is. It's just an endotoxin found in the outer membrane of gram-negative bacteria. So they have receptors for that. They have receptors for mannose as well. And once they pick up these components, like the receptors bind to mannose or bind to LPS, then we hyperactivate the macrophages and they become even better at phagocytosing. And so they get really greedy and they start gobbling the pathogens more avidly. And they also produce now and secrete some cytokines. So they will be secreting interleukin-1 and tumor necrosis factor alpha, which allows other cells to know, okay, there's a problem here. But in itself, something like TNF alpha, for example, it can kill tumors and cells that have been, let's say, infected by a virus, okay? Now, hyperactivated macrophages also produce reactive oxygen species. You've heard me talk about these reactive species, these molecules that have unpaired electrons in the outer orbits, and it makes them highly reactive, right? So these macrophages do that because the reactive oxygen species can help kill pathogens. Very useful. And that's why, sometimes people say we should just get rid of ROS, or reactive oxygen species, right? But we actually need it. It's part of the immune defense because that's one way that we can kill pathogens. And the other thing that the macrophages do, they generate a lot of ROS to kill pathogens, but they also can release some of the lysosomal contents on any invading pathogens, okay? So in a nutshell, very, very quickly, we talked about the phagocytic capabilities of macrophages, but they are also secreting cytokines like interleukin-1 and TNF-alpha, and they are antigen-presenting cells because they're going to gobble up bits of what's going on in the environment, let's say the pathogen, and then display some of it on the MHC2 platform to inform cells of the adaptive immune system, to give those cells information about the enemy, okay? So there we have macrophages, very, very generalized. Now, I just want to briefly mention dendritic cells, dendritic, dendritic cells. And these I consider sister cells to macrophages because they can pretty much do what macrophages do, but they have sort of a different interest, okay? So dendritic cells, they can phagocytose, right? They have that capability, and they are also able to present antigen. We think of dendritic cells mostly as professional antigen presenting cells, okay? And in fact, their phagocytic ability is mostly so they can just take in bits of the environment. So you can think of them as sampling the environment. What's out there? Let me just take a little bite of what's outside there. And when I sample that, whatever I find, I can display it on my MHC2s, right? And present that to the adaptive cells, okay? So we think of the dendritic cell more as an antigen presenting cell. It's able to phagocytose, but it does so primarily to sample rather than to just go after and kill the pathogen, okay? So those two are done, macrophages and dendritic cells. Let's move on to neutrophils. Okay, neutrophils, you think of, macrophages, very, very sort of complex, and they have so many roles. There's a killing role, but they also have intelligence gathering. They also stimulate, T cells and so forth. They're secreting cytokines. Neutrophils, very simple. All right, mostly they're there to kill. Okay, they're very destructive. They generally live about 5 days in your bloodstream, and they're a lot of times just hanging out in the bone marrow. And when activated, they can really get to the site of infection very quickly, within an hour, all right? And mostly what they're going to do is phagocytose the pathogen, secrete some cytokines like TNF-alpha, again, to tell people, there's a lot of problems going on here. Now, they are kind of indiscriminate killers because they're just going to basically destroy anything in sight, right? So they're going to phagocytose, but they also do something very interesting. They release neutrophil extracellular traps. Neutrophil extracellular traps, and the acronym is NETs, or capital N-E-T, and then, the plural NETs, okay? So NETs. And when I think of NETs, I think of Spider-Man, because Spider-Man, he's always shooting out these web strands, right? And you can think of the neutrophils as doing that, which I think is just amazingly cool. So they're like spurting out, these nets, sort of as a suicide gesture, because they don't survive very long after they do that, okay? And I'll tell you why. Okay, so they're squirting out these nets like Spider-Man is sending out these strands of his web, except you can think of these strands as barbed wires, okay? So with the neutrophils, the barbed wire, the wire part is made up of their own DNA. So they're just basically squirting out their own DNA, and the barbs are these very toxic proteins that will destroy pretty much anything in sight. Okay, so it's got this barbed wire web that it's just kind of spitting out into the environment, and that process is called netosis, as they're generating the nets. And as you can see, if you're spitting out your own DNA, you're not going to live very long, right? Because yeah, that's not a good thing. Okay, so the idea is that these nets are going to kill the bacteria that are around, for example, because I told you those proteins, those barbs attached to the DNA, those are very toxic. But the other thing that it can do with the nets is it's literally like a net. So we're going to trap bacteria, right, to that area. You don't want bacteria floating around, going to other parts of your body. So it's useful to kind of hold them in that area, limit that area where they are, and then focus on killing them, okay? So that's what neutrophils do. Now, how do they get to the site of infection? It's like most white blood cells. How do they get to the site of infection? Well, they're generally just, obviously they're in your lymph, but eventually they go into the bloodstream, and then they're flowing by pretty quickly, kind of merrily flowing by in the bloodstream. Okay, when you have infection, then endothelial cells, remember, the glycocalyx is going to get degraded in some of those areas where we've had infection, okay, or inflammation. So, the macrophages are there. They're sending out interleukin-1 and TNF-alpha, and we know that that's not a normal situation. So the glycocalyx gets degraded there, and it's going to reveal what? These molecules, adhesion molecules, that the endothelial cells have on them. Generally, they're hidden by the glycocalyx. But now as we degrade the glycocalyx, we start to reveal some of these adhesion molecules, okay? So now we have the neutrophils and other white cells that are going by merrily along. But as they're doing so, we have these other molecules that the endothelial cells will express that kind of bind to the neutrophils, okay? And now instead of having the neutrophils flowing by very quickly, they kind of slow down and they start to roll along the wall, the lining of the blood vessel. So instead of zipping by, they're now slowing down. They're at the lining of the blood vessel, and they start to roll because we have these sort of sticky molecules that are binding to those neutrophils and slowing them down. Now, when they get to the sites where their adhesion molecules are, sort of revealed, then they actually have proteins in them called integrins, integrins, and these integrins can bind to the adhesion molecules, and now they stop at that site. Okay, so that's where they stop. They were rolling slowly, and then they have their integrins bind to the adhesion molecules, and they stop. And at that point, they can then enter into the wall of the blood vessel and then go into tissue. So that's kind of how they get to sites of infection. All right, now let's move on to NK cells, or natural killer cells. All right, so you can know from the name, they're really good at killing, or they wouldn't be called natural killers. They basically are able to live about 7 days in your bloodstream, and also in the liver and in the spleen, which is where you can find them. At the sites of infection, they secrete cytokines like interferon gamma, which is a type 2 interferon. We'll talk a little bit about that later. And they're able to kill tumor cells and virus-infected cells and bacteria as well. And the way they do that is they actually force the bacteria to self-destruct, okay? What they do is they are able to punch a hole by using perforin proteins, PERFORIN. So again, I told you, like punching holes, right? So they punch a hole with perforin proteins, and now then they actually dump in lots of toxic enzymes that will destroy the cell. So that's essentially how they like to kill cells. The other way they can do that is they can bind to pathogens. So they have something called FAS ligand, FAS, and ligand, which is really the recognition site. And they are able to bind to FAS, FAS, on the pathogen. Okay, so FAS ligand will bind to FAS, and then now basically we can initiate a program to get that invading cell to destroy itself. So how do natural killer cells know who to destroy and who to kind of leave alone, right? So back to our MHC platforms. See how important they are, right? Most healthy cells in your body, you know, we have in our cells the MHC1 platform, and all the time we're posting information about what's going on inside the cell. And we're just sending out signals, yeah, business as usual, right? Because, you know, as you have different cellular processes, you're degrading proteins and so forth. And those are all normal. So you post on your MHC1 and, you know, everything's fine. So when your NK cells, natural killer cells, are looking and they see that you have that platform going, you got some MHC1 posting going on, things are probably good. Okay, so that's an inhibitory signal to natural killer cells not to destroy that cell because they're posting MHC1, you know, information, and I think everything should be fine. Now, sometimes though, let's say a virus gets into a cell, and now this virus is able to infect the cell, be inside the cell. Now, if the cell now knows this, it tries to destroy the virus that's inside. Maybe, you know, it tries to put it in a lysosome and destroy it, right? It'll digest as much of it as possible. And some of these viral bits, it can now post on MHC1. And this is very important because as they're posting these viral bits on MHC1, we have these T cells from the adaptive immune system, and they're looking around. And when they see these posts on MHC1 in that cell, wait a minute, that's an invader, right? That's not a normal part of that cell. Yes, we're posting on MHC1 what's going on inside the cell. And what we're seeing is that there's an invader inside the cell. And then the T cells will kill that cell. Okay, basically to limit infection. So it's very important that way. However, viruses are very smart too, and after all, they want to survive. So when they figure that out, they go, well, okay, what if we don't allow the cell to post on MHC1? So we'll modify it, mutate that cell that we've just infected, and they won't be able to have MHC1, and they won't be able to post about what's going on inside, and no one will see us in here, okay? So it's all nice and quiet and secretive. Okay, the natural killer cells now are looking around, and they see a cell that doesn't have a lot of MHC1 posts. Maybe very few MHC1 or none, right? Instead, it's got some weird platform that's kind of put up there just to kind of distract you, okay? So you might see some strange carbohydrates or proteins instead of the MHC1 proteins. Okay, when we see a lot of that in the cell, the NK cells, the natural killer cells go, okay, we're going to get rid of that cell. So that's a kill signal to the NK cells, right? And it's going to go to that cell and make it self-destruct because it realizes that something's not quite right. Okay, so once we remove the MHC1 postings, the T cells can't look in there, but the natural killer cells will go, wait, why are there no MHC1 postings, and we've got this strange other platform there instead? That's probably an infected cell, and we're going to kill it. Okay, so you can see there's a little bit of backup system going on there. All right, so now let's take a step back and look again at the innate immune system and consider that it's a ready-made system to defend your body. It's not specific to any antigen, but when it senses that something's not quite right, it springs into action to try and defend your body, right? And the way we do so is with pattern recognition receptors, or PRRs, all in caps, okay? Pattern recognition receptors, there are about 20 of them. We can broadly divide them into two groups. One would be PAMPs, PAMPs, again, all in caps. And these are pathogen-associated molecular patterns. Pathogen-associated molecular patterns. And they recognize things like, again, LPS, the endotoxin, right, on gram-negative bacteria. So that's one group. Then we have DAMPs. DAMPs, all in caps. DAMPs. These are damage or danger associated molecular patterns. Okay? And these are intracellular molecules that are released by damaged or dying cells. And they kind of signal to the immune cells something's not quite right here. All right, so the most well-known of the PRRs, or the pattern recognition receptors, are the toll-like receptors. TOLL-like, toll-like receptors, and we can just refer to them as TLRs, all in caps. All right, and different cells have different combinations of TLRs. The most famous TLR is probably TLR4. That was the first TLR elucidated by the famous Charles Janeway, who won a Nobel Prize for that. And this TLR4 is on the cell surface, okay? And what it can do is it can sense certain things, such as LPS. It can sense viral proteins, for example, heat shock proteins. It can also sense LDL. It can also sense fatty acids like palmitic acid. Okay, so there's certain things that it recognizes as toxic, and they're very generalized. It's not like, oh, it's recognizing a specific bacteria, but it's recognizing, you know, bacteria that express LPS on their outer membrane. Okay, so generalized in that sense. And once they do that, we activate a program in the cell to actually bring about an inflammatory response. We start the inflammation process, okay? So we need the sensor. The sensors are the toll-like receptors, in this case TLR4. And once the sensor is activated, we run the inflammatory program. Now, I told you TLR4 is on the cell surface, but these TLRs can be in other parts of the cell. So take, for example, TLR7 and TLR9. They are actually inside the cell, and you can find them in phagolysosomes. All right. TLR7 recognizes single-stranded RNA. So like your HIV virus, right? That single-stranded RNA, that will be recognized by TLR7. TLR9 recognizes double-stranded DNA, like in bacteria, for example. Now, remember, they're in the phagolysosome. So once we phagocytose the pathogen, right, and then now we fuse with a lysosome and we try to like destroy it, as we break up the pathogen, right, and then we have bits of DNA floating around. If it's single-stranded, TLR7 will pick that up. If it's double-stranded, then TLR9 will pick that up. And then again, we activate a program in the cell to rev up the inflammatory response, okay? So I just want to stress they're not specific, but they're so powerful, these pattern recognition receptors. And I think one of the coolest things about them is that they basically recognize vital structures or features on pathogens that are, you know, just mandatory for that pathogen to survive. So there's part of the LPS, okay? We need that in the outer membrane of those gram-negative bacteria. Now, if we try to modify it, it's likely the pathogen itself won't survive. So it kind of has no choice but to express that, right? And these PRRs recognize those vital structures that are very hard to mutate. And then they can set about that inflammatory response. Okay, so those were the pattern recognition receptors. Damage, I just want to point out the DAMPs. Okay, anytime we see a lot of damage in the area, right, and maybe something's not quite right, and we see a lot of dying cells, a lot of, destruction, tissue destruction, right? Again, that is a signal that something's not quite right. And, we can alert the immune system via that kind of PRR. All right, let's very now briefly go to the interferon system for countering viruses. So interferons, they're called interferons, INTEFERON, interferon. Okay, it's called interferon because it interferes with viral replication. That's where the name comes from. So when you're attacked by viruses, then your cells secrete interferon alpha and interferon beta. These are type 1 interferons. So, so let's take fibroblasts and macrophages. Okay, when you're invaded, they secrete these interferons, but at the same time, they have interferon receptors on them. And the interferon that they secrete, they're going to bind to as well. And then what they're going to do is they're going to make a lot of antiviral proteins to kind of decrease the viral load that way. The other thing it will do is they're sending out this interferon into the environment so that nearby cells, when they pick up on the interferon signals, they're going to go, uh-oh, there's trouble here. And we're going to prepare to activate our death program. Okay, so it's going to prepare to kill itself in case it gets invaded. So the interferon is a signal to neighboring cells. All right, if you get invaded, you should just self-destruct to limit the spread of the virus. Okay, very smart that way. Now, if that cell doesn't get invaded, then obviously nothing happens. It's not going to self-destruct. But if it does get invaded, then it's been warned, it's been primed, and it's going to set up its own self-destruct program so that we don't let the virus spread. And the cells that make the most interferon would be these plasmacytoid dendritic cells, plasmacytoid. And really, they are able to detect viruses via TLR7, TLR9. They start pouring out interferon to try to limit the spread of those viruses. Okay, so we did a very, very general survey of the innate part of the immune system. Let's quickly do our wrap up. We started off by just defining the innate as the ready-made, very quick to act system. The adaptive one takes a longer time to rev up, but it becomes very specific to certain pathogens, okay? Now, in the innate system, we talked first about the complement proteins and how they can kill viruses via assembly of the MAC, or membrane attack complex. They also tag the viruses for destruction by phagocytes, and that process is called opsonization. And so they actually act as opsins to tag the pathogens that way. And they can act as chemoattractants to bring other immune cells sort of back up, right, to the area. Then we went into the phagocytic cells. We talked, of course, about macrophages. Now, macrophages, they do a whole range of things. So we know of them as phagocytes, and they are very good phagocytes, but they can also present antigens. So when they're primed properly, they become antigen-presenting cells. And this way, they can gather information from the battlefield, act as intelligence cells, and bring that back to, let's say, the lymph nodes to educate and inform the adaptive immune system. Okay? Then we did talk a lot about the MHC platforms, right? They're kind of like social media platforms that the cell has on the cell surface. MHC1, you can post about what's going on inside your house, right? So inside the cell. MHC2, you can post about whatever's going on in your environment. So if your neighbors are doing something, someone down the block is doing something, you can post those things, all right? So that's the broad category of the MHC1 and the MHC2. So when the macrophage swallows something up, chews it up, breaks it up, it can take bits of whatever it just engulfed and display it on MHC2, because after all, it swallowed it from the environment, came from outside, okay? And that will eventually inform T cells, how to become specific in looking for that pathogen. Okay. Then we talked very quickly about the sister cell, the dendritic cell, which can also phagocytose. And at the same time, though, it mostly is an antigen presenting cell. So when it takes in bits of the environment or bits of the pathogen, it's kind of sampling and taking little tastes of what's going on outside in the extracellular environment. And then it can take that information and present it again to cells of the adaptive immune system. Right, so that's dendritic cells. Then the other killers we talked about are the neutrophils, and specifically these are the ones that can do the netosis process. So it's kind of like Spider-Man shooting out the web, and the web in this case is made up of the strand of DNA from those cells, the neutrophils themselves, that are laced with these very toxic enzymes that will kill the bacteria. Now, the nets also, besides killing the pathogens, will limit them to an area so we don't allow things to spread too much. But, basically that's a very destructive process, and we end up destroying a lot of our own tissue as well when neutrophils are on the battlefield. We talked about natural killers, and natural killers essentially basically do the job by getting the cells that the pathogens to self-destruct. Okay, they recognize by actually looking at certain signals that cells display, and those signals are essentially through MHC1. So they're going to be looking and seeing if you're posting anything through MHC1 about what's going on inside. If you are, then likely things are okay, right? If you're not doing a lot of internal postings, and instead you have some weird platform with strange proteins and strange carbohydrates instead of those MHC molecules, then that's a signal to the natural killer cells to go after that cell and shut it down. Okay, then we talked about pattern recognition receptors, which is how your innate cells and your innate immune system is able to recognize that there's trouble going on. They cannot specifically identify a certain pathogen and target a certain type of bacteria, a certain type of virus, but they know when something is not right. And they have these pattern recognition receptors. We have pathogen-associated molecular patterns, and we have damage-associated molecular patterns. Those are the two big general groups. The most well-known are the toll-like receptors, or TLRs, and then the most famous is TLR4, which is on the membrane of the cell, so it recognizes what's going on in the extracellular environment, right, and then sets up a signaling cascade to basically activate an inflammatory response in the cell. Now we have TLRs inside the cell as well. So TLR7 and TLR9 are in phagolysosomes, and when they detect bits of pathogens in the phagolysosome, if the pathogen has single-stranded RNA, TLR7 will sense that. If they have double-stranded RNA, then TLR9 will sense that. And basically, again, we set up an inflammatory programming in the cell that we activate at that point. Okay, so those are the pattern recognition receptors. And then finally, we just briefly talked about interferon and how interferon really interferes or interrupts viral replication, and interferons also are very useful in signaling other cells of the immune system that something's going on, and priming cells of the immune system. Okay, so a very brief, really, overview of the innate immune system. We kind of swept through that, but think, at least we have all the main components described and discussed, and we can go into depth later. But more importantly, now we can talk about disease processes, and I can talk about these different components of the innate immune system, and you'll have an idea of what we're talking about and what those components do. Okay, so again, please give a like if you enjoyed this episode or you are getting something out of this podcast. You can also sign up for my newsletter on vivianlohmd.com, VYVYANELOHMD.com. For now, signing out from VLMD Rounds, I'm Dr. Vivian Loh, and I sing the body electric. Bye.